Assalamualaikum warahmatullahi wabarakatuh. So for the first chapter, we are going to go into introduction to biotechnology. So in this chapter, we are going to look at several things, generally the things that you are going to learn, or at least get a gist of information on what we are going to learn in this class. So in this figure, we have a figure of biodiesel, DNA, tomato injected with something, and then some machine. And also you can see that probably the plant is connected to a device. So things that probably are going to be explained to you in this class. Okay. So biotechnology, there are so many things that comes to people's mind when they talk about biotechnology. Mainly the things that probably boggle their mind is DNA. Anything that has to do with DNA, anything that has to do with genetic modification, so things like that. So we're going to go deeper. So what are biotechnology? But I'm going to focus more on agriculture, since this class is is created specifically for students in the plantation industry. So biotechnology is actually anything that has to do with living things, and such as we have plants, we have animals, we have microorganisms, viruses. In fact, viruses are not considered real living animals, real living organism, but in fact it is a parasite which has to be dependent on something in order for them to live. So for example, if you have coronavirus, which are specific to human and probably swine and monkeys probably, or probably pangolin. So if there isn't any presence of these animals, so they actually cannot cannot live, because they have no suitable host for them to live. Okay, so when we say bio is any living thing, and technology is the technology that we use to manipulate the living things in order to make them beneficial or useful, or for anything that has to do with human life and consumption, or probably things that can ease your life. For example, this guy, it's a hand sanitizer. It says it kills germs. So the function of this is to kill germs, but how do you produce this thing? So it says ingredients is isopropyl alcohol. So the first ingredient is 70% alcohol. So where do all these alcohol comes from? Actually, the alcohol is coming from fermentation, which is where you add microbes to sugar, and you give them an anaerobic condition so that they can produce ethanol, and then you can collect them. So the amount of alcohol that you can collect it depends on what kind of fermentation that you do, but mostly there is if let's say it's 100-liter tank, so probably in 10% of them, or probably 10 liters of them, are contributes towards the production of alcohol. Okay, so even these are also a product of biotechnology, because the simplest thing, which about the same application of this one, is such as the making of wine. For example, the ones that we have in Shah Alam, or the Carlsberg factory, which uses a big tank. So that big tank are actually fermentation tank that they can get the product of fermentation, which is ethanol, by collecting from the tank. So they have a valve that you can collect all the ethanol once it has formed through the fermentation. Okay, so there's also another example of biotechnology. Another thing that there are so many things that you can think of, but these are the simplest idea that you can get. Hold on a sec, I'm going to show you another example. I have right here some kind of a shampoo or a shampoo or what we call uh, soap, okay, or foaming bath or something like that. So sometimes there are things in this thing which we call it as enzyme, which does not really produce much bubble or foam when you wash it. When you wash your hand, you wash your body, it doesn't really produce that much of foam. So so some people might think, so what's the point of using that soap or that bath, because it doesn't produce that much of bubbles. So they feel it's not really that clean because there isn't so much bubble produced with when you use it. So actually that's not always true, because sometimes there are also soaps that incorporate, not soap, but for example dishwashing detergent, probably the detergent that you use for your washer, which has less foam, especially the ones that you have with the front-loading washing machine, which has much lesser foam in it, because they don't use much water. So that's why if let's say you have to use a washing machine that has a front-loading washing machine, you are not supposed to use too much. You're not supposed to you're not supposed to use just any type of detergent. You actually need to use the one that produces lesser amount of bubbles, because this type of washing machine does not use much water. So probably what comes through your mind is, when it doesn't have so much bubbles, so how do it cleans? Actually, these guys, they produce, it is made from enzyme. Okay, it is made from enzymes which can target the dirt on our shirt. For example, every day we sweat. So we sweat. We're probably some people might be we call it, or probably some people, they shed. I'm not sure what kind of shedding, but they probably shed their skin off, or probably some of the anything that has to be with coming from their body that they shed, or they being flushed by the body through your minerals or your sweat or something, anything that is contained in your body. So all this is got stuck to your shirt. And sometimes when you eat things like oily food such as sambal and everything, they have lots of oils associated with it, and then it becomes so hard for you to remove it. So those who produce soaps or detergent, they observe that actually the dirt are actually coming from oils or probably proteins. So what comes through their mind is these things can actually be targeted by lipase and proteases. So by knowing that lipases and proteases can target lipid or oil and proteins, so what we can do is, by producing soaps and detergents, then can actually target this protein and lipid, so that they can cleave the lipid so that it gets broken, and eventually cleaning your clothes. Okay, so that's how it works. So that is enzyme. So how do we obtain this enzyme? Is by looking at nature. So probably you need to do some research. Probably what you need to do is pouring the oil onto some places and see if it becomes decomposed. So most of the time decomposing will cause the smell of the oil to be rancid. So the rancidity tells you that actually some microbes are actually present. So you can collect and isolate all this bacteria and see which one reacts with oil or eats oil. That means they use oil as their food. So these are the kind of bacteria that you can use to produce those enzymes. So when you have already identified this bacteria, you can either take the gene from this bacteria and reproduce it into E. coli, another bacteria that is easy for you to culture. So now we have a genetically modified bacteria, which we can culture in many in bigger flask, in bigger tank, so that you can have lots of the enzyme, because we can isolate it later when we have already cultured the bacteria. So that's another example of biotechnology in general. Okay, so just as you see in the in the first slide, you can see that there is a tomato. Okay, there is a tomato that has been injected with something. So actually we have already have a lot of tomatoes which has been genetically modified, where the issue being is tomatoes have short shelf life. So and another thing is if we pluck it when they're not ripe, so the taste will not be as tasty as it is when you pluck it when they're not ripe, when they are ripe, sorry. So imagine two things. This is a tomato that you pluck it when they are not ripe. They are almost ripe. That means they are still green but having some red color on it. And another example is the group of tomato that you collect when they are already ripe. That means they are sweet. So this ones you're going to sell. Both of them are going to be sold to the shelf in the market. So basically, when someone buys it, the ones that is ripe will taste more sweeter than the ones that aren't ripe, because they have already got enough maturity, enough sugar breakdown when they're on the tree. So this one contributes to the sweetness of the tomato. But the ones that you pluck before it ripes has less sweetness than the ones that you pluck after it's ripe. So what we want is, we want it to be still, but another issue is the ones that are ripe will rot faster because they are ripe, and so they will decompose faster. But the ones that are not as sweet will stay longer in the shelf, but it has compromised in its taste. So what we want is actually the ones that have a longer shelf life but doesn't compromise on the taste. So that's what we want. So what we do is, by manipulating the ethylene gas production, which comes back to the gene, by modifying the gene that controls ethylene expression, we can actually control the ripening. That means you can pluck it when it's ripe, when it's sweet, and then we can sell it to the market while still retaining its sweetness and taste. Okay, so those are the examples of biotechnology that I can give you. So in other words, biotech is any technological application that you can think of, that you can apply to living organisms, living things, to improve them so that it can be useful for human use. Okay, so that it's more than just a single technology. In fact, by knowing if someone has a COVID or not, that is also considered as biotechnology, because you take the people's sample and then you do DNA testing. So DNA testing is basically part of part of biotech application. Okay, so for example, in medical biotechnology, we have diagnostics. There are two examples or three examples that I can give you. Number one is pregnancy test. Okay, so for pregnancy test, if someone gets pregnant, how do they know that they're pregnant? This is by looking at all those. There's a strip, pregnancy strip, which when you pee on them, through chromatography, your urine will travel towards the sample testing. So it will tell you whether you are pregnant or not. Okay, So actually, when you're pregnant, they learn that your body produces a hormone called a hormone gonadotropin, which gets produced by your body by only pregnant lady's body. So that's why they know how accurate that kit is. Okay, by peeing, by urinating on that strip, and it'll travel. It will tell you whether you're pregnant or not. So that's one, because it detects the protein that is present in your urine. And another example is by HIV test, which usually those who are going to get married, they will undergo this test in order to tell whether they have HIV or not. So through this, it is also the same concept, which is they take a look at the presence of something in your blood, so they are able to tell whether it's present or not. So usually the concept of this one is by a lock and key concept. For example, this is your body represent the key. The test strip represent the ones that are going to your keys that is going to go into. Okay, so if let's say your key fits into that knob, that means you have it. But if your key doesn't fit into that knob, so that means you don't have it. You cannot open the door. Okay, so that's how it works. Okay, it's like an enzyme substrate concept. Same thing. So if it's present, that means you have it. If it's not present, you don't have it. So that's the thing. Another example I can give is coronavirus. So you know that the viruses are composed of two things, which are protein, and another one is RNA, the RNA. So in this type, so every living thing has a unique composition of protein and RNA. So when it's unique, that means you can detect it through its difference. For example, you know that is your friend with the name so-and-so by looking at their face. You know that this is certainly your friend by just looking at their face. How do you know it's her? I just know that it's her because her face looks like it is her. So same thing goes to this one. By looking at your protein sequence, so we have a sequence, DNA sequence, protein sequence, and all. By being able to recognize specific sequence that is unique, that belongs only to you, that tells it that it is actually you and it's not someone else. Okay, so that's how we determine what is who is who and what is what. Okay, so they take your nasopharyngeal swab up to here, and why it is there? Because the virus lives there. Okay? It's just the place that where it lives. And so they collect the sample. So what they do is they can do PCR. PCR means polymerase chain reaction, which detects the sequence of the RNA virus that is present in your sample. Okay, so they can tell if it's present, then it will show a band. It will give an indicator that tells you that it's present. Okay, so all this marker, the indicator or the marker is present in your body. For the case of pregnancy, the indicator is the hormone, which is produced in your urine. Okay, if you have dengue fever, so how do you know you have dengue? It's by looking at all this marker, which is produced by your body and not produced by someone's healthy. How do you know you have cancer? It's by looking at all this marker that is produced only by a disease, a cancerous person, which is not present on someone who's healthy. Okay. Next one, therapeutics. So therapeutics also work about the same concept with diagnostic, which if they know this is the one that causes the disease, so how do you turn it off is by binding to them. So it basically shut it down, and it shut it down, so it no longer causing diseases. Okay, that's therapeutics. For example, they thought, that's one of the examples. Other examples that doesn't have to do with binding is, for example, you have the ones that you put yourself in the chemotherapy, sorry. So the chemotherapy works by killing cells. So when you have cancer, cancer means when your body starts to divide uncontrollably. So you want to be able to control. It's actually, this chemotherapy does not help you to control the division, but it's just that it's killing the extra cells that you have so that it will not cause any harm to your body. So when they detect any excess growth in your body, they will it will kill that specific cell, and eventually hoping that you will recover because all of these cancer cells gets killed. So that is also in a form of therapeutics. But most of the time that I learn especially me as a biotechnologist, most therapeutics focus on targeting that specific disease. For example, if let's say, like I said before, if this is the disease, so how do you work to turn it off so that it shuts it down, it doesn't, it no longer expresses the disease anymore. Okay, for example, there are so many examples. Okay, for example, let's say you have an allergic reaction. You eat something, or probably you were bitten by something. You're bitten by some weird insects that you have never been bitten before. So your body starts to produce all this allergic reaction, and your face becomes swelling. You have inflammation all over your body because your body is responding to it. It's giving response by performing inflammation. So this inflammation is actually because your body is trying to get rid of that thing. So it swells and it inflames all this. So if let's say you have a vaccine, or you have the vaccine will try to, or actually the medication would be to turn off the histamine. So usually the ones that causes allergy is histamine. So how do we deal with histamine? By turning off the histamine by giving antihistamine. So that means if this is the one cell that causes you to be ticklish, itchy, and everything, so they produce like a switch to cover it. Okay, so that you no longer feel the itch, the burn, the everything. Okay, they turn off the sensation by covering your ticklish sensation. Okay, so that's how by targeting and clothing. Same thing happens. Same thing goes to Panadol, or the scientific name is paracetamol. Okay, what's paracetamol do? Well, actually, paracetamol is a type of a placebo, which doesn't really cure. What it does is only it desensitizes your sensory. Okay, so basically, this is the part that you feel that is painful and having diseases. So what it does is it desensitizes, meaning it no longer feels sensitive towards something. So when it desensitizes, you no longer feel the tickle, the ticklish, the itch, the pain, the burn, the whatever you think. Okay, so that's how it works. Okay, so vaccine. Meanwhile, for vaccine, if let's say a virus is causing you to be having a fever, so they take just a little bit of that sequence of the virus. They cut it down and put into something else, and they reintroduce it into your body. So they will weaken that virus and reintroduce it into your body. Okay, so that your body will develop antibody towards it. So next time if you get infected by the same virus, you will be okay, because your body builds up memory against it, so it knows, it remembers who has already been in your body. So it just, if let's say next time if someone which has the same character or traits like the one that has been injected, so they recognize it, so they just attack it right away. So you get basically you get you can become um, okay by a few days only, as compared to those who have like 14 days or 11 days of sickness and illness. Okay. Next one, we move on to agricultural biotechnology. For plant agriculture, like I said before, we have tomato that has been incorporated with genes that has been manipulated. It is actually the gene from itself, but it's just that the gene that produces ethylene has been cut or manipulated. Sorry, not cut. Manipulated so that it produces ethylene in a slower fashion. So when it is produced in a slower fashion, it will not make your tomato raw as fast as it has always been. Okay, and that's one example. There are other examples, such as you can make a resistant. You can also make resistant tomatoes, resistant against diseases, resistant against herbicide, or anything that you can think of. That whatever that comes to your mind, you just name it. You can just do it if you know the theory. Okay. Next one, for farms. You can create fertilizer of your choice if you think it's good for you. For example, there are so many fertilizers that that uses microorganisms. So they created through biotechnological knowledge, so biological knowledge. So they know that this plant probably this plant would produce much more fruits and all if you incorporate certain types of fungus into its root. So you create a type of fertilizer that contains all these microorganisms, which helps them to supply materials, supply nutrients to the roots, and also at the same time prevent them from getting the diseases. So all this is also some of the branches of agricultural biotechnology. And you can also, like I said, if let's say you think that your plant is it is hard for you to kill the weeds around your plant because the weeds are omnipotent. Omnipotent means it's present everywhere. So when it's present everywhere, it's hard for you to control it. That means it's not wise for you to use pesticides. So next time, what we do, there are also examples of plants that are herbicide resistant. That means you can spray all over without killing your plants. That means wherever you don't have to worry about killing your own plant. You can just spray whatever, but your plant survives. Okay, so that is herbicide resistant. For food processing, like I said, you have wine, you have the making of tempeh, which has about the same idea. And also, how you make tempeh is actually the white thing is a fungus. So the way you make it is by by inoculating new fungus into your soybeans. Let's say you have new soybeans. I have never done it before. This is a theory that I thought should work. If let's say you don't have the fungus, what you can do is take the tempeh that you already bought. So you take a little bit of that fungus and put it to your soybean and then wrap it and just leave it there for 2 days probably. So it should grow in 2 days. The fungus should grow in 2 days, and you should find your soy ready by that time, your tempeh ready by that time. So that's how you make tempeh. How do you make yogurt? Same thing. So the only thing you need are milk and yogurt, because the yogurt contains the bacteria that will turn your milk into yogurt. So you need yogurt and milk. So you dip let's say you get a spoon and you heat it so that it sterilizes your spoon, and you dip it into your yogurt and you dip into your milk, and then you stir and you leave it in somewhere warm for 2 days, and then you can eat it. Okay, well, probably you can just pour your yogurt into your milk about in several proportion. So stir it and leave it into a warm place, and your yogurt is ready by 2 days. So that's how you do it. Okay. Next one, environmental biotechnology, cleaning through bioremediation. So this is very, very important, because if there is no bacteria to help us clean the world, so your world will be dirty. Okay, so we have swines, which works in a larger scale, and we also have microorganisms that works in a smaller scale, micro scale. Okay, so for example, you have just eaten a bowl of spaghetti and you can't finish it, so you just throw it on the ground, hoping that someone will help to clean it. It is actually someone does clean it. Someone actually clean it. So how do they work? They produce enzymes. So these microbes, they will colonize your food, and then they produce enzyme and breaks your food, and then the nutrient is returned back to the soil. Okay, so these scientists, they have observed on what kind of bacteria that can actually help them to clean the world. So they found out there are bacteria that eats oil. So these bacteria, because they eat oil, so they were applied to the ocean when there were events of oil spill in the ocean. So they just spread the microorganisms. They will help them eat, and they help us clean the ocean by eating and breaking the oil. Okay, this is made possible, like I said, by the production of lipase. All right, that's one example. There's a lot of other examples you can go and figure. So, and preventing environmental problems, like I said, same idea is by cleaving, by producing enzyme, by cleaning it. So when it's cleaved, so they get decomposed naturally. Okay. Next one, forensics biotechnology. So how do you know if some people is wondering if they are the true parents of yours? Probably you're adopted or something. So you want to know if that's your real parents. So what you can do is paternity test, taking the sample to the sequencing of the DNA people, and they will do with PCR and check it for you and see whether it matches your parents. Okay, so that is for a paternity test. And that's one. If, let's say, someone dies in about a month and they become disfigured badly, so because they have disfigured, so only some samples are present for you to check on to see who this person is. So you take that sample, you get someone else's sample in order to match them. Then you run the test and see if it matches, if it has the same band. Okay, so that's how we do. That's among the things that you can do in forensics biotechnology. And also for scientific investigation, probably what comes to my mind is you can Google HeLa cells, H-E-L-A cells. I forgot the real name is. It's probably Henrietta Lex. Okay, so this lady has already died like 50 or 60 years ago. She died of cancer. And however, although she has died, she's a black lady, African American. So it has been very controversial. So what happens is her tumor cells have been collected without the consent of the family, and the cancer cells were taken from this lady, dead lady, and currently are used as samples to study cancer. So I had that chance to use HeLa cells when I was doing my degree. So basically, I'm using some dead lady's cancer cells, which has died 60 years ago. Okay, so the cell is still alive. Okay, so that's for scientific investigation. Next one, industrial biotechnology. You have energy production. these are some of the things that you can produce at home, and it's really amazing and interesting. For example, you can just buy, if let's say you have used cooking oil, you can turn them into a biodiesel by just reacting them with lye. Lye is sodium hydroxide and methanol. So you can go find the YouTube on how to produce biodiesel by just having three things: lye, which is lye means sodium hydroxide, methanol, and your oil. That's all. That's all. That's all you need. And probably the next, the purification method will take a lot more effort. But the basic needs, the basic things that you need are those three. Sorry. Okay, so why do we need biotech in agriculture? Is basically to increase yield to feed everyone in the world. So we don't, because we have much less and less land for us to plant nowadays, because of urbanization and increase in population. So we want to fight that situation by producing more yield so that people can eat. Okay. So the real disease is hunger, not have enough food at affordable prices. So sometimes you are not that hungry, but your issue is being malnutrition. For example, you don't really have much time when you're working, so, and you have 5 kids at home, and you don't really have time to cook. So the easiest way for you, if let's say you go home late, you get home late, so the easiest for you to do is to go to drive-through, McDonald's, KFC, and get some food, bring it back. So it's not really hunger, it's actually malnutrition. So these are the issues: hunger, malnutrition, all those things are combined, and those are the issues that we want to tackle by using biotechnology. Okay, so these are some of the examples of biotech in agriculture: multiple insect resistant cotton, GM rice, transgenic mustard, GM brinjal, herbicide tolerant onion, transgenic maize, stress tolerant rice. So all these, let's say, stress tolerant rice, if let's say you cannot live on a place which has too much water, or probably cannot withstand drought, so you can actually manipulate them in order for them to be able to live in a robust condition and in a climate changing condition. So how do you do it? This is very basic. Actually, it's not so hard once you understand it. You have, let me show you. So this is your bacteria of interest. If let's say you know that this bacteria produces lipase, so you can extract the DNA and take a piece of the sequence that correlates to what you need because in the bacteria it has so many genes that correlates to whatever that is supposed to do. For example, this will help you to produce something, this will help you to produce something. So you just take the ones that you need, and then you clone it and design the genes, you incorporate it into another host, and you can transform it back into your plant and culture it and breed it. Okay, for example, if let's say that bacteria can produce vitamin A. Okay, so in this case, so you can cleave the gene, put it back into that plant gene, transform it, reculture it, and here we go. You have a full-grown plant. Okay, so for bio other applications, you have seeds, breeding, genomics, seed and market value. So you can do biochemistry. There are so many things that if you go and if you just learn it, go read and watch YouTube. There are so many ideas that you can do if you take biotechnology as your field. Okay, you can change the DNA. You can produce product that you want. Anything. Just you have to think more creative about what you want to produce. Um, some of the history. So basically, the ancient people, they observe and selected superior breed of crop, and they start to observe that actually plant can actually interbreed and produce better plant. Okay, so and then it grows even before Christ, 4,000 before Christ. So it then proceeded and improves over time to production of microscope. There was discovery of antibiotic, and until the production of genetically modified organism. Okay, so and so 1985. This is the first genetically modified plant, which is tomato, which, like I said, they have modified modified its ethylene production to produce a longer, prolonged shelf life, shelf life. Okay. A lot of other things, and if you go over and read, and you also you'll probably have heard of Dolly, which is the first cloned mammal. And nowadays you have the latest was the Nobel Prize winner of 2020, which is the ones that has the idea on gene editing. Gene editing. You can go and look for gene editing, CRISPR, which has already produced babies, human babies, which can be edited, whose gene can be edited. So the name is Lulu and Nana. Lulu, a Chinese baby, which is very, very controversial. Lulu and Nana actually their gene is actually HIV resistant. Okay, so you can go and read for Lulu and Nana. These two girls, these twins, are HIV resistant, and the parents are HIV positive. So basically, these two couples, they got married, and they thought of having child. So what they do is they went to see a scientist, and they produce child, and they edit the gene of the child for not being able to get HIV. So basically the baby is HIV resistant. So this is produced through the method called CRISPR-Cas9, which this CRISPR-Cas9 is initially discovered from bacteria, and they discovered how bacteria react against viruses. So how the bacteria kills viruses when they are infected. So they discovered CRISPR, which can edit the gene, which the bacteria deletes the virus from their body. So they use this application to Lulu and Nana. Okay, so why do we need biotechnology? There are so many things. I cannot name it because there are so many. If you read, and the question will be everlasting. There'll no full stop for this question. So many things you need it for. It's for life. It's for the world population. It's basically for the people, for the world, and everything. For sufficient food, fiber, and shelter. And also, this is to overcome the issues of deforestation, overgrazing, and land conversion. We no longer have enough spaces to grow plants, so we have to figure out how are we going to increase our yield and at the same time feed the whole population in the world. Okay. So this is a quick recap that I've talked about. You have plant revolution, the use of biotechnology. You have animal science, medicine, food technology, environment, all these. And also we have genetically engineered animals, in fact humans, plants, animal vaccine, which for example insulin initially comes from swine sources. which they get because swine has the most similar insulin like humans, so that's why we took it from the swine. So this we reculture the insulin gene, so it's coming from the pigs. Okay, so we produce insulin and then we can inject it to human who has problems with diabetes. Okay, and then we have plant protoplast fusion, which allows you to combine two things, two protoplasts of different types. You fuse them, for example, you and your brother. You take your skull, you combine, and then you become you and your brother, twin brother, whatever. Okay, so that is to fuse two things together. Okay, by combining the nucleus. Next one, embryo transfer. For example, if your wife keep on miscarrying your baby, so what you can do is, if let's say you have lots of money, you can pay someone to be pregnant for you, for your baby. That means you have the sperm and the ovum of you and your wife. That means the zygote, which of you and your wife. You can transfer it into someone else's womb. That means a surrogate mother, and call it a surrogate. Okay, so this is embryo transfer. So let's say the baby is from test tubes, so you can have a surrogate mother to help you to carry your baby and give birth to it. And improve microorganism, recombinant DNA for disease diagnostic, monoclonal antibody production. Monoclonal antibody production is to produce medicine for you to fight certain types of disease, antibody, for example. So antibiotic. I'm sorry. So we have always been fed with antibiotics. So these antibiotics is coming from from the process called monoclonal antibody production, and a lot of other things. And so the examples, the things that we did, that agriculture do, emphasize rice and wheat production, tissue cultured planting materials, breeding through conventional or genetic engineering, high yielding crops with short rotation for cultivation. So all these are, just like I said, the summary of what I've just told you: better plants, better variety, more yield, more plants with smaller spaces, fertilizers, and biocontrol, which pesticide free, chemical pesticide free, or inorganic pesticide free. Okay, and also diagnostic kits, which you can detect diseases of that plant. We're just using kits instead of investigating of what kind of diseases they have been infected with. And we have genetic engineering, which is the technique that transfers gene of interest to develop and improve plants, animals, and other organisms. So it goes back to your genes. So every living thing has a gene, which is a basic physical and functional units of heredity, which carries information for the expression of a particular trait. So when someone says your face is similar to your mother's face, so it is because you have her gene. But actually you have both. You have your father's and your mother's gene. It's just that your mother is more dominant than your father. So the gene is actually the one that dictates how the product is going to be. So your nose is probably, it's either like your father or your mother. If it's different, you gotta check whether everything is different or just one of the things that is different. If everything is different, that gives you some doubt that you need to find out, to discover whether you are really their child or probably foster child. So in this figure, we can see the conventional breeding and genetic engineering. So in conventional breeding, it takes a long time for you to accomplish, with no definite answer whether it's going to happen or not. But for genetic engineering, you choose that specific trait that you want and you incorporate into the host that you want. So it should display the kind of organism that you want. So conventional breeding is limited to exchanges between the same or very closely related species. It can only happen between banana and banana, not banana and pineapple. But in genetic engineering, it allows you to take enables pineapple and banana to take place. And for conventional breeding, there is little or no guarantee of obtaining any particular gene combination from the millions of crosses generated. So you were throwing 1,000 balls into a big pail, okay, a big pail. You expect that you can grasp the specific ball of your interest, which is not always going to happen, because it's going to happen only by chance. Okay, so this will take a long time in order for you to get the desirable combination of your intention, of your yearning. Okay, but for genetic engineering, it's not it is possible for you to do it, because by just identifying the gene of interest, cut it and put it and transfer into your host. So that's, it's just simple as that. It sounds that simple, although it's not that simple, but actually it's much simpler than conventional breeding. And undesirable genes can be transferred along with desirable genes, which takes a long time to achieve desired results. Like I said, genetic engineering takes only specific gene of your interest, but doesn't happen with conventional breeding. Okay, so some of the examples of GMOs that you might have eaten a lot, such as soybean, squash, corn, potato, and I can't see the ones in the back. I don't know how to move it. It's okay. And global area of biotech corp. So there have been so many countries nowadays that are developing biotech. So in fact, in Malaysia, we are in short of biotechnologists. So these are the examples of transgenic crops, or GMO. You have bananas, papayas, corn, coconuts, and wheat. Types of okay, so the benefit is we should have improved food quality, food processing with better tasting, more nutritious, cleaner food. The use of enzymes and bacteria. Actually want better food, which is more healthier and saves the people from getting processed food. And we hope that everything will become better by using biotechnology. And also, if let's say you're dealing with animals, we want it to have leaner meat, more milk, and better production. For insulin, for diabetes, you can also have, for medicine, you want to produce lots more other medication for people, to treat people without using any chemical intervention, a lot of synthetic stuff that we want to feed the people. We want it to be naturally occurring and actually targeting the disease. And then we have a bioindicator. Bioindicator, for example, when I was in my degree, I did isolation of luminescent bacteria, which is a bacteria that produces light when it's dark. That means the name of the bacteria is Vibrio. I forgot what the name is, but basically it's luminescent bacteria, which can only be found in the deep ocean. But what we did was, the project was to find this luminescent bacteria, which is in the deep bacteria. What we did was by buying shrimp in the market, and we soak it in salt water, so that we collect the bacterial sample from the salt water, which is the bacteria has fallen and become soaked in that water. So we collect it and isolate the bacteria. So eventually we are able to see that it glows in the dark. So it produces light when there is no light. Okay, so that is so this bacteria actually has a bioindicator, which allows you to see if you need light. Okay, probably, and you can take that gene, which is the lux gene, which produces less fluorescence. You can use it for by indicator, put it somewhere else so that it can become a marker. Okay, so that's it for today. If you have any question, you can ask me. I'm more than happy to answer your question, and I hope you enjoy the lecture today. Thank you.